In the last lecture, we developed a couple of mathematical models that could be used to describe the growth of populations of organisms. These were the exponential growth model, which assumes that populations can grow infinitely large and never run out of resources, and the logistic growth model, which acknowledges that there's a carrying capacity, which is the maximum size a population should be able to achieve given the amount of resource available in the environment in which it lives. Now, we know that populations sometimes experience exponential growth, but only under certain conditions, such as when a population colonizes a new environment, or when a previously large population has been reduced to a very small number and is recovering. What about logistic growth? Do natural populations actually follow the S-shaped curve we described last time that's predicted by the logistic growth model? This is the question we left off with last time, and this is where we're going to pick up our discussion of population dynamics today. After we answer this question, we're going to go on to look in a little more detail at the factors that actually regulate the growth of populations in general. Well, does the logistic growth model describe the growth of real populations? The answer to this question is yes and no. For example, consider a population of a single-celled pond organism called Paramecium. You may be familiar with Paramecia since they're commonly found in any freshwater body or even in a puddle of water, and they're a favorite subject of amateur microscopists and of high school biology classes. You can easily grow a culture of Paramecia in the laboratory. All you need is a small flask or even just a test tube of water that includes a supply of food. For example, if you just throw in some rotting vegetation, the bacteria and other things that serve as food for these Paramecia will grow, and you can use that to develop a culture of Paramecia. Now, if you start to sample a population of Paramecium that's growing under these kinds of controlled conditions, you typically observe that the actual growth curve of a Paramecium population very closely follows the S-shaped curve that's projected or that's predicted by the logistic growth model. Specifically, you see that there's initially a slow rate of increase, but as the population size builds, this rate of increase starts to itself increase, and the curve begins to have a steeper and steeper slope. But then at some midpoint, as the population reaches its theoretical carrying capacity or approaches its theoretical carrying capacity, then what happens is that the curve starts to level off. The slope of the line begins to decrease, and eventually the curve will approach asymptotically the carrying capacity of the population. So at least under these kinds of controlled laboratory conditions with fairly simple organisms, we can see that a population's growth will nicely match the predictions of the logistic growth model. We also observe logistic growth in wild populations of organisms. For example, there's a population of fur seals that lives on St. Paul's Island, which is off the coast of Alaska. This population was hunted extensively during the 19th century, almost to extinction. Beginning in 1911, however, hunting on this population was banned. There were only a small number of individuals left, but none of them were being killed anymore. And at this point, wildlife biologists began to monitor the size of the population on a regular basis. And they did this over the next several decades. When you look at the growth of this population, again, you see that the population actually very closely follows a curve that you would expect from a logistic growth model, with a gradual increase that becomes more and more rapid until finally, as the population reaches the expected carrying capacity, the curve eventually levels off. And then after a period of time, you see a relatively constant number of individuals living, about 10,000 breeding males in the case of this particular population. Well, we can see logistic growth then in the laboratory and in the wild, but not all populations really closely follow a logistic curve, even under controlled laboratory conditions. For example, consider population growth in another common pond organism. This is actually a small microscopic multicellular organism that we call Daphnia, or its common name is a water flea. Again, you can grow populations of Daphnia very conveniently in the laboratory and follow their population growth. If you do this, you often see with Daphnia that the population begins to follow a growth curve that would be predicted by the logistic growth model. But in fact, instead of leveling off when it approaches carrying capacity, the population size will overshoot the carrying capacity. In fact, it might overshoot it by quite a bit. Now, once the population has gotten to a level, to a density that is far above the carrying capacity, then what happens is the population will come crashing back down again. In fact, sometimes it will go then below the carrying capacity and then gradually come back up. In fact, populations like this might even oscillate once or twice around the carrying capacity until finally they reach the carrying capacity that's predicted for this population and level off and maintain a relatively steady population density. Now, this pattern of population growth is actually commonly observed in populations of many different kinds of organisms. In other words, the populations will eventually reach a carrying capacity, but they don't do it in the smooth, continuous fashion that's predicted by the logistic growth model. Now, the fact that the logistic growth model doesn't exactly predict the way real populations grow shouldn't be too surprising to us. After all, it's just a relatively simple model. The fact that it doesn't, however, means that there must be some kinds of assumptions in this model that really are not met in natural populations. And there's one assumption in particular that we can identify. This assumption is that the potential negative effects of increasing population size as a population reaches its carrying capacity would have an instantaneous effect, an immediate effect on the population's growth rate. In other words, our model suggests that as the population increases bit by bit, there will be an immediate feedback on the population growth rate, which slows it down so that it'll approach its carrying capacity gradually. In fact, there's often a delay in the response of a population's growth to its approach to its carrying capacity. For example, what you might find is that a population is limited by the amount of food available. As the population's size approaches carrying capacity, we would expect that the decrease in the amount of food available per individual would slow down the growth rate of the population. But many individuals will store resources, basically resources that they can use for further growth or further reproduction. And so the fact that they're not immediately getting the same amount of resources won't actually slow down the population growth rate right away. The overshoot occurs because these organisms will be using stored resources to continue their reproduction. And only after these stored resources run out will the population eventually adjust and become closer, with its size coming closer to the carrying capacity that we'd predict. In fact, this situation likely accounts for what we observe in species like Daphnia, which do store resources that they use for their immediate reproduction. Well, the logistic growth model of the logistic model of population growth, I should say, predicts that once a population has reached its carrying capacity, it will maintain a relatively stable population size. In other words, whether it reaches that carrying capacity gradually or in some more less gradual fashion by overshooting it, once it eventually gets to its carrying capacity, logistic growth suggests that it will no longer change its population size. And the reason for this is that our logistic growth model would predict that the reproductive rate of the population, R, would become zero. And effectively it becomes zero because of that term we added to the equation last time, k minus n over k. Once n equals k, that term becomes zero, and so we predict zero population growth. Now, in some cases, we do observe that populations maintain a relatively stable population size once they reach a carrying capacity. But in many other cases, we don't even observe a stable population size at any point in the trajectory we might see of changes in the population over time. Instead, many populations will fluctuate in size, increasing at some times and decreasing at others, and they may never appear to stabilize at a carrying capacity at all. These population fluctuations really can be quite dramatic. For example, in a population of Darwin's finches that have been studied by Peter and Rosemary Grant in the Galapagos Islands, you may remember we talked about some of the Grant's work earlier in the course when we were talking about the evolution of beak sizes in Darwin's finches. Well, the Grants have been monitoring populations of different species of Darwin's finches for decades. And in one particular species, they found that over a 20-year period, the size of a particular population of these, particular population of a species of these finches, the so-called cactus finch, would vary from a low of as few as 50 individuals to a high population size of as many as 500 individuals. In other words, that's essentially an order of magnitude difference in the size of this population. Now, even populations that fluctuate in this way often do so within some established or regular range of a minimum and a maximum population size. So we have a couple of situations here. We have some populations which reach a carrying capacity and remain relatively stable. We have some populations that fluctuate relatively radically, and we have other populations that will fluctuate within some established range of a minimum and a maximum population size. These kinds of patterns that we observe once we assume a population should have reached its carrying capacity lead us to ask the question, what is it that actually regulates population size? What are the factors that actually determine changes in the growth rate of a population that will lead to these changes in size? We have a theoretical model, the logistic growth model, and that's the best model we've come across so far in our discussion, that suggests that reproductive rate should go to zero. The net reproductive rate of the population should go to zero as the population size approaches its carrying capacity. But what actually causes reproductive rate to go to zero? What other factors cause reproductive rate to increase or decrease even though the logistic growth model might suggest it should be zero? Well, I've already told you that in the most general sense, the factors that affect the reproductive rate, the growth rate of a population, involve resource limitations that affect birth and death rates of individuals in that population. Now I want to take just a little bit of a closer look at how these kinds of mechanisms actually work. Population ecologists separate the mechanisms that might regulate population size into two general categories. These categories are referred to as density-dependent effects or density-independent effects. Now, density-dependent effects, as the name suggests, are those effects that intensify as the density or population size of a population increases and that reduce as the density of a population decreases. They're density-dependent effects. By contrast, density-independent effects, again as the name suggests, are factors that affect a population size irrespective of what the current density of the population is. Let's take a look at how these factors work in turn. First, let's take a look at how density-dependent effects regulate population size. Actually, the logistic growth model we've been talking about really is explicitly a model of density-dependent population regulation. As the population approaches its carrying capacity, the increasing number of individuals in the population divide the resources available ever more finely on a per individual basis. As the amount of resource per individual decreases, then in general we would expect the per capita birth rate also to decrease, and we would expect the per capita death rate to increase. All right, so we have a decrease in the birth rate, an increase in the death rate, and that's why this net factor that we've talked about, the population growth rate, R, would eventually go to zero. Or in fact, R might become negative if the death rate exceeds the birth rate. Now, interestingly, this response of population growth to population density is an example of negative feedback. This negative feedback operates in essentially the same way as we saw negative feedback operating earlier in the course when we introduced the concept of homeostasis. Now, the difference between this kind of negative feedback and negative feedback in homeostasis is that the mechanisms involved here haven't evolved explicitly to regulate population density. They're just factors that happen to have a negative feedback effect on population density. But nonetheless, it's an example of how change in one parameter, say a population increasing or decreasing relative to some set point, the carrying capacity causes a feedback mechanism to return the population size, the factor in this case, the population size, to its normal set point level. Now, In addition to the availability of resources that we've been talking about per se, just for example, the amount of food an individual may eat, there are a number of other factors that link population density to population growth rate. For example, there may be competition for less obvious resources, things like nesting sites, or space needed for territories for animals, or a suitable substrate for something like a plant to grow in or a sessile marine organism to attach to. Predators also may be attracted to areas where the population of their prey is most dense, and in fact may avoid areas where the population density of their prey is less dense. In this case, the effect of predation will be increased with increased population size, and this then would represent another example of a density-dependent factor that, in this case, increases death rate. Similarly, diseases may be more prevalent, more readily transmitted as populations become more dense. So as population size increases, the prevalence of disease may increase, again increasing death rate. In fact, one last kind of an example of a density-dependent factor that's not directly linked to resources per se is simply the accumulation of waste products. As the density of a population increases, whatever waste products they produce will also increase, and the occurrence of these waste products may have some negative feedback on the growth rate of that population, decreasing births or increasing deaths. Now, it's fairly easy to demonstrate density-dependent effects on population size in natural populations, and it's sometimes even possible to identify which particular factors are specifically operating. For example, a population of song sparrows, these are small birds, field-living songbirds, the song sparrows living on an island called Mandardi Island off the coast of British Columbia, has been studied for many years by Jamie Smith and Peter Arcese. Again, they've been monitoring the population of song sparrows on Mandardi Island and asking questions about how various other life history parameters change with respect to population size. This population also varies in size quite dramatically. In some years, there can be fewer than 20 breeding adults on the whole island, and in other years, there may be over 150 breeding adults. Again, quite a broad range of variation in population size. Now, in this population of song sparrows, Smith and Arcese have been able to identify several density-dependent factors that appear to have a direct feedback on the growth rate of the population. For example, the number of males in the population who fail to obtain a territory increases as the total number of males in the population increase. Now, this isn't very surprising. It's an island. There's limited suitable habitat for territories. As the number of males increases beyond some point where all males can get a territory, some males simply fail to have a territory. Now, in a songbird like a song sparrow, the inability to get a territory for breeding essentially means that that male can't breed at all. So in this case, we see that the inability to obtain a territory is a density-dependent factor that will have a negative effect on overall growth rate of the population. Another example of a factor that seems to regulate this population of song sparrows involves the observation that as the number of breeding females on the island increases, the average number of offspring produced per female decreases. So as more and more females are breeding on the island, each one of them actually produces fewer young per year. Presumably this effect is due to resource limitation. As there are more females trying to raise young, trying to find food to raise those young, there's less food overall to go around, and so the number of young that actually make it out of the nest and survive to adulthood goes down. Finally, Smith and Arcese have been able to show that the probability that a young bird, a bird born in 1 year, the probability that that bird will survive over winter to the next season also varies with population size. As population size overall increases, the probability that the young birds will be able to survive their first winter decreases. Again, this is presumably because the increased population density makes it more difficult for those young birds to gather the resources they need to be able to maintain themselves through the more difficult period of the year, which is the winter on this island off of British Columbia. All of these density-dependent effects that Smith and Arcese have been able to identify provide examples of negative feedback loops connecting population size to the overall growth rate of this population of song sparrows. Well, let me give you another example that demonstrates how density-dependent factors work in general to regulate population sizes. This is kind of an interesting example. It involves a species of fly that form what we call galls on goldenrods. Now, goldenrods are common field plants. You're probably familiar with them. And this fly, like many insects, actually has lays its eggs on the goldenrod, and then the young larvae of this species of fly. This species of fly, by the way, is called Eurosta. This is the genus name for the species. It's also used as the common name for the species. So these Eurosta flies will lay their eggs on goldenrod. The larvae will burrow into the goldenrod stem, and the presence of the larvae in the goldenrod will form what we call a gall, essentially an expanded nodule in that goldenrod stem that creates a protective environment in which that larvae will live and grow and develop, feeding on the resources provided by the goldenrod. Now, Eurosta flies are widespread in goldenrod fields, and you typically find them occurring in relatively low population densities, and you typically find that their population densities remain relatively constant. This suggests that there must be some factors that closely regulate the population size of Eurosta flies. Now, what could those factors be? Well, one possibility is the presence of resources. For example, enough goldenrods for those flies to lay their eggs on and have their larvae feed on. Another possibility is the presence of predators that themselves might feed on the larvae. So these galls are fairly obvious things in a goldenrod field, and it turns out that there are a number of species of predators, including things like parasitic wasps or birds, that will home in on these galls and actually use the larvae living in them as a source of food. So wasps might burrow in and lay their own eggs on the larvae, using the larvae as a food source for their own young, or birds might peck their way into the gall and in so doing pull out the larvae as a tasty little treat. Now, it turns out that it's been possible to show that it's not resource limitation per se, but instead specifically predation that appears to regulate the population size of Eurosta flies. And the way that this was demonstrated was through the following kind of an experiment. Eurosta flies were taken out of natural goldenrod fields. All of the flies were collected. They were collected actually in the form of galls. After the pupae lived through a summer, or after the larvae lived through the summer, they form a stage in an insect's life called a pupae. And this pupae will overwinter, and then then adults will emerge from these pupae the next year, and the adults will then go and mate and lay their own eggs. So researchers would gather these goldenrod galls, bring them into the laboratory and hatch out all the adults. Now, what they've done is they've removed adults from natural fields. They've hatched out these adults in the lab, and then the next spring when these adults emerge, they will go and replace adults into fields. But they'll do this with varying the density. They'll replace the adults in some fields at the same density in which they normally occur. But in other fields, they'll replace them at a much lower density. And in yet other fields, in a third treatment, they'll replace these adults at a much higher density than they normally occur. Now, what is observed when this kind of experiment is done is that in higher density plots that have a higher density than normally are observed in the field, in nature, all of the adults that are put into that field will be able to lay eggs and actually have their larvae colonize goldenrod stems and create galls. In other words, you'll see a higher proportion of galls in these goldenrod fields where you've increased artificially the number of adults in that population. This result suggests that it's not the number of goldenrods that's limiting the population size. There appear to be plenty of goldenrods to go around. But what you do see is that the percentage of galls that are predated varies with population density, varies with the number of galls in a field. Specifically, if you have relatively few galls in a field, then you find that a low percentage of those galls are actually predated by things like birds or wasps that are getting the larvae. But as the number of galls in a field increases to a size that is beyond what is normally the density of a population of Eurosta flies, what you find is that the percentage of galls that are predated increases to a much higher level. This change in the percentage of galls that are predated with the change in density suggests that predation is a density-dependent factor affecting population growth in Eurastra flies. As the density of these flies increases, or the gulls they produce increase, the percent of them that are actually attacked by predators goes up. Presumably the mechanism here is that the predators are homing in on fields where they have a higher probability of finding a gull because there are just more gulls there, and they're avoiding fields where there's a lower probability of finding a gull. So this is an example then of a density-dependent effect that is regulated by predation, affecting the regulation of this population of insects. Well, let's turn now and look briefly at density-independent factors and how they may affect population size. Now, by definition, the occurrence and severity of density-independent factors affecting population size is unrelated to population density. It's density-independent. Often, density-independent factors have something to do with weather or climate or other kinds of disturbances to the habitat. For example, the first freeze in the fall will often have a major effect on the survivorship of many insect populations. When a freeze occurs, many of the insects in a population will simply die. They don't survive the freeze. But the percentage of insects that do die is unrelated to the density of the insects in that population. Whether it's a high density or a low density, the same percentage of insects will die because of the freeze, a density-independent factor. Let me give you an example quickly of one system where this kind of density-independent effect on population size has been documented. Now, this example comes from another insect. This is an Australian insect, or an Australian species of a kind of insect called thrips. Now, thrips, again, is a genus name, and it's also used as a common name for this insect. Thrips are insects, small insects that feed on pollen or leaves or flower tissues of plants. In fact, they often feed on roses and other cultivated flowers, and so they're considered a common pest species by gardeners. Now, what you see if you monitor the population size of a population of thrips is that during the winter, now this is the Australian winter, so this would be in what we would consider the summer months, say of around June. During the winter, the temperature is fairly cool. This cool temperature actually impedes the development of individual thrips, and as a result, individuals don't mature fast enough to reproduce before the flower on which they're living itself dies. The fact that the flower dies before the thrips die, and the fact that before the thrips reproduce, and the fact that the thrips are unable to reproduce because the temperature is cold, is an example of a density-independent factor that keeps thrip populations to a very low level as long as the temperatures are cold. They'll never approach a carrying capacity because they simply can't reproduce fast enough when they can't develop fast enough. Now, with the onset of spring, the Australian spring, say, being in November, the weather warms up, the thrips are able to develop at a faster rate, and in fact they do start reproducing. And what you'll often see in a thrips population with the onset of spring is a very rapid increase in population size, something that actually approaches what looks like exponential growth. But this exponential growth doesn't last, because as soon as the temperature gets to be too hot, then what happens is that the excessive heat actually causes the thrips to increase their mortality. They can't take the dryness associated with the hot Australian summer, and now the heat of the summer increases mortality of the thrips population, and the population size comes crashing back down to another low level. This is a second kind of density-independent factor that's controlling or regulating the size of a typical thrips population. Now, the important point to make here is that these thrips never seem to even have a chance to approach their carrying capacity, because one factor or another, cool temperatures in the winter or hot temperatures in the summer, act as density-independent factors that keep the population size from ever growing large enough to approach carrying capacity. Well, in many populations, both density-dependent and density-independent factors will act together to determine how a population changes its size dynamically. In many cases, populations might remain near or fluctuate around their carrying capacity, but periodically have their population size greatly reduced by some density-independent factor. And then once the population size decreases, then as the logistic growth model would suggest, the population will slowly recover, approach its carrying capacity, being regulated in this case now by density-dependent factors. It's hard to imagine any population, if it approaches its carrying capacity, would not be regulated by density-dependent factors. It's also hard to imagine any population that isn't potentially regulated, at least at times, by what we've called density-independent factors. By identifying cases where we can isolate density-dependent factors that regulate populations and density-independent factors that regulate populations, we can gain insight in how resources do, or in some cases don't, help explain the abundance and distribution of populations of organisms, which, as I told you before, is really a central question we want to answer when we're talking about the ecology of organisms.